Hello? We'll be right there. Guys, there's been a murder. A forensic scientist is someone who helps investigate crimes by collecting and analyzing physical evidence. Most scientists specialize in either crime scene investigation or laboratory analysis. At a crime scene, forensic scientists will typically walk through the scene to determine what and how evidence should be collected. They will then take photographs of the crime scene and the evidence. They will then collect all relevant physical evidence, including weapons, fingerprints, and bodily fluids. They will then catalog and preserve evidence before transferring into a crime lab. Forensic scientists may use tweezers, blacklights, and specialized kits to identify and collect evidence. In addition to processing crime scenes, they may also attend autopsies.
So this is our blood sample that we've collected, and now I'm going to use it to test the blood type of the victim. When scientists identify blood types, they rely on slight differences in the antigens or protein markers on the surfaces of red blood cells in a blood sample. In a body, those antigens are recognized and attached to by antibodies. If antibody proteins detect red blood cells with foreign antigens, they attach to those antigens and cause them to clump. Forensic scientists often use techniques to identify blood types because an individual's blood type isn't affected by disease, drugs, climate, occupation, living conditions, or any other physical circumstances.
In the process of gel electrophoresis, DNA fragments are loaded into a gel and placed in an electrical field, which sorts the DNA fragments into various bands. These bands can be colored with a radioactive dye to make them visible to imaging techniques used in electrophoresis. The colored bands created in this process have a specific pattern that corresponds to only a certain individual's DNA. Forensic specialists can see if a pattern produced from gel electrophoresis from one person's DNA at a crime scene matches with another person's, and can use this process to eliminate suspects or identify victims.
After running the gel electrophoresis test, we discovered that the DNA found on the hair of the victim is the same DNA as the victim has.
The only explanation for the correlation between the DNA of the hair and the victim is that the victim and the person belonging to the hair on the victim are related and actually twins because they have identical DNA.
Now, we're looking at fingerprints, which were found on the victim's neck. Our victim was strangled, so we know that those fingerprints were from the murderer. And I'm looking at them now in the microscope. They're definitely not the victim's. And so, knowing that there was her twin present at the scene, now I'm going to use these fingerprints, run it in the database, and they match her twins, as I can see on the screen.